this session. So please be aware that this session is recorded. I see that all of you guys, all of your, for the most part, all of your, we have about 10 participants. All of your videos are off and all of your audios are off. I'm going to be sharing a document. When I share the document, I'm basically sharing a whiteboard and talking a little bit about this lab. I can't really see the group chat when that happens, or I don't notice when things are in the group chat. So if somebody could be monitoring that and actually tell me about that. How can you do that? Would you be able to do that for me to tell me, just unmute and then tell me? Again, I need get a little feedback from you guys. I'm talking from basically my basement here. All right. Good morning. Good morning. Yeah, so just let me know if somebody puts something on chat. So hopefully all you guys are doing okay. Well, you wouldn't say anything if you weren't doing it.
There are like 12 of them in the group.
I'm sorry, what, Tran?
There are like 12 people typing in the group chat.
Yeah, okay. Well, I've been typing a lot inside of there, so we'll do that. So basically, so I am recording. So what I'm going to do is I'm going to go on to share. And I have a whiteboard that I'm going to do on share. Does everyone see my whiteboard?
Yeah, I see it.
Okay.
Oh, quick question before we begin.
Yes.
The last lab, were you going to send us more data about the planaria, or should we just finish the lab without that data?
You know what? I'm going to try and get the data. So Christian is actually able to go into the lab. I'm not able to get into the lab. They locked me out. Security will kick me out if I go there. Christian is going to go there, and hopefully he will get one last picture of the planaria. Basically, they should all be healed by now. So remember before we had the head and the tail section? I took 2 planaria and cut them in half. We should have 4 planaria now, 4 full planaria that are a little bit smaller than they were before. So I'm hoping to have that for you guys by Wednesday. If not, then I will send you a note. I would like to have all of these things by midnight on Friday. That way you guys could start your spring break with everything all done, for me at least. Is that okay?
That's the same day of the exam, so it'll be a little crunch, but.
Would you rather me do it than on Sunday?
I personally would, because I needed time to focus on the exam, because it seems like I was trying to get the specifics from Dr. D about what the exam's going to cover, and he was a little vague. So I know I'm going to spend a lot of time studying on that. Okay.
So yes, so I will go ahead and change that. Thank you. This is Nelson saying that.
It is. Yeah. I hope others feel the same way. It's not just me.
Yeah. So I kind of didn't want to go into your spring break, but if you're willing to do that, I am okay with that too. And I realize that some of you will be kind of focusing in on that. So I could take that, these last few labs I'll put a summary up, and I will take them on Sunday night by the end of Sunday. So midnight Sunday night.
Thanks. I really appreciate that.
Yeah. Okay. All right. So kind of pop in with your questions here, and I'm going to go into this. So basically, we're looking at blood group determination. So this is lab number 30. It's a relatively simple lab. It shouldn't take you that long to do. I have examples out there, and I'll give you that in a little bit. We have some hard labs that are coming up on mitosis, meiosis, and things like that. I'm hoping we have some simulations that are actually pretty good that I'm hoping we get the license for those simulations so that you can do that. So it's kind of like virtual reality with these simulations. And so you'll be able to do those hopefully a little bit later. But for right now, we have the labs. Was everyone able to go out and do the go out and have a field trip and take a look at things outside?
Yeah, I haven't finished it, to tell you the truth.
Okay. But, you know, again, that will take you about an hour. You'll need to sit outside, just sit and look for about 20 minutes, probably about 3 times in 3 different areas. So you should be able to do that.
Yeah, I see a lot of people in the group chat saying they did it.
Okay.
Melissa did it. Jonathan did it.
Okay. All
Poe
right.
did it. Gerald.
Well, that's good. Yeah, I got a really bad case of poison oak doing that. So it's kind of laid me up for a little bit. So what I wanted to do on blood group determination, so what this is looking at is your RBCs, red blood cells, okay? The red blood cells are an interesting type of cell, right? Because they are responsible for transport transport of oxygen, okay? And so you can, if you bleed a lot, if you get a very severe wound, you can bleed a lot and you can lose these cells. Now, if you can't transport oxygen, what's going to happen to the organism fairly quickly? I don't know if you can see my video on here, but you're going to die relatively quickly. And so what we as a human species realized relatively early on is that if we can take excess blood from other people and put them into different people, that this would actually save a fair amount of lives. Trouble is, when we did that, some really bad results happened. And basically what happened was a process that we call agglutination. So and that's from your book, AGG. Actually, that's a little bit faster. And specifically with red blood cells, we have hemagglutination. Hemagglutination, where the red blood cells. So what I want to show about this is that what I want to do with this group here is to show that RBCs have antigens red blood cells have antigens on them, and antibodies bind to antigens. And so basically what we have here is shown is that we have basically 3 major kind of blood groups. So the ABO is the A has these A antigens on here. The B has these B antigens here. The D has these basically is lacking those antigens. So I put out these little spikes out here, but they don't have that. A regular cell doesn't have the red blood cell antigens. The reason is because they are not red blood cells. They have their own antigens. And so let me put a drawing on here. And I can put a drawing on here. And so again, your red blood cell, your regular cell has these own antigens, okay? And so basically what's going on is these antibodies. So an antibody, and so if I was to draw an antibody, so an antibody is a structure that kind of looks like a Y. It's too bad that I put on a Y, but we have the heavy chain and the light chain. But this is an antibody. An antibody is basically a protein that's made by a vertebrate organism, and it's basically the vertebrates that have this. Invertebrates don't have this as well. This vertebrate is made to have basically a portion of it here, color, and this has a portion of it that actually is sticky and actually will bind to an antibody. So if I take an antibody, so here's an antibody, and actually let's erase that. If we take an antibody and let's do this one blue. And so what we see is we have some I believe we have some anti-A antibodies. I'm going to draw format blue. We have some if we have some antibodies, so let's say I have an antibody. So this is the A antibody. And I'm going to draw another B antibody. B antibody. And so what's really kind of interesting is this A antibody will actually can I select? This A antibody will actually bind with the A antigen on that one. The B antibody will actually bind with the B. In both of these antibodies, because there's nothing there for them to bind to, they can't bind to the O. Also, because there is no antibody on a regular cell, these antibodies can't bind to it as well. So when we do this, we're actually able to, what we're actually able to do is we're able to determine, what the blood type is. So I'm going to save this. Okay, all right, so this saved it. Okay, cool. So if I go in here, I'm going to just erase everything. Okay. Does anyone have any questions so far? Not hearing from anyone, so it's.
Oh, I had a quick question. While I was reading the chat, it says something about individually use the template. So is this a individual lab, or can we do it in groups? Or
Yeah, it's an individual lab. Let's do it individually.
individually? Gotcha.
Yeah. So what I will do is, so I'm gonna. So you guys will have. Sorry, this is my Google document. So here's your Google document, Biology 215, right? And so this Google document will be in lab number 30, blood group determination. Kavya already did it. So here's the copy of the template with all this in here. And what I'm going to be asking you to do is what I've done, for, read the lab book. And so here what I've given you is I've given you 3 actually pictures from my class last year that did this. And so we have a you, which pretend that's you. We have the person on your right, pretend that's your person on your right. And we have the person on your left, that's your person on your left. And what we're going to see here is some results. And I will kind of pull these up. And so what you're supposed to do is when you have the anti-A, and that's kind of the blue one. The blue is the anti-A, and that's why I blew the antigen A. You want to look for hemagglutination. And I'm going to talk about hemagglutination in a little bit. And then what you're going to do is on here is then you're going to give us what your phenotype is. That's what you look like, and what your genotype is. And I will talk about that in a little bit as well. And then you're going to answer these questions, and that should be it. It should really take you, I'm figuring, about 30 to 40 minutes to do this particular lab. So you don't necessarily need a lab partner. Is that okay?
Okay. And then after, I think if you were showing us stuff, the screen was just white.
Okay, let me. Okay, let me. Okay, so here's a screen right here. The screen is white right now.
Yeah, it's white. Yeah, it's just white.
Okay. All right. So what I wanted to do is I'm going to draw on this screen.
Okay.
So what I wanted to do is basically draw. I want to draw agglutination. So here are a bunch of red blood cells, right? And so if we were to take a, do you see the cells on there or no?
Yeah, I see that now, the little red circles.
Okay. And so if we were to take a look at these red blood cells on a, so normally what we would do to do a blood type, so we would put three little, you would put three little boxes or areas on a slide. You would then put a drop of your blood on that slide, and so the blood in that slide, and then you would put an antibody in here. And so when you put that blood on the slide, it gives you roughly a red, hazy color, right? And that's due to thousands of these red blood cells being kind of all over the place. And I'm trying to do that right now, right? So if we put all these red blood cells all over the place, it would give you kind of a hazy color. And again, that's what this kind of looks like. Trouble is your red blood cells, so let's make these A's. Your red blood cells, so this is A-type blood. If we put in our B-type antibody, so that's B, this is not to scale, please. Okay. Nothing happens, right? Because these guys are all, nothing really happens with these guys because the B antibody doesn't bind. So I'm going to go through and I'll erase out the B. The trouble is when we put in the A antibody, which is blue, it actually, it will actually bind to, I'm going to select it. It will actually bind, it will actually bind to the red blood cells. And a lot of times these red blood cells have many antibodies on them. Not antibodies, so antigens on them. So what happens is that I'm going to draw another. So this is the A antibody. And so when you have, so I'm going to try and, So this A antibody, this will bind here. You put in another A antibody. And what happens is it actually binds to these red blood cells. And this is what gives you the hemagglutination. And so what we see here is those red blood cells are now kind of all clumped together. And I can't do as many in there. And so what we see is that's what it looks like. So that's hemagglutination. And so what I will do now is go back to my template. And if I go back to an older class, spring 19, this is lab 30. Okay, here, Ibrahim, I don't think I used his. So here's, can anyone take a look at this? Is there any hemagglutination going on here?
Hemagglutination. I don't even know what the fuck that is. Ooh. Well.
I mean, we could even try and magnify it a little bit. Right? So there's this kind of hazy red.
Nick.
I'm sorry, what? Are you seeing it?
We can't see it, no. We're just seeing the same drawing you just did.
Okay. All right. So let me go share. All right. Do you see it now?
Yes, there you go.
All right. So this is looking at, so what this is this is a slide that has the anti-A antibody, the anti-B antibody, and then the anti-O. And you can't see it there for some odd reason. Let me get that a little bit smaller. All right, so if you take a look at all three of these, basically a drop of blood, this individual's blood was placed in there, and that this blood was mixed up with the antigens. And do you see agglutination?
No.
No. So what's the blood type of this individual going to be?
O negative?
That's exactly correct. Ding, 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 ding. Yes, O negative. So let me go back and see if I can take a look at some other students. These are not some of the better slides, and actually that's one of the slides that I used for class. So here's a good one. So what do you think this individual is? Do you guys see it? Nelson, do you see it?
Yeah, I do see it, but I really don't know what I'm looking for. I think.
Okay. Do you see my cursor going around here?
I do, yeah, around B.
So around B, A and B is what's happening is that's the agglutination, right? So this is your Rh antigen. When you don't have the cells agglutinating, you will have this general diverse, diffuse red blood cells, right? And so, and this is here, you're looking at agglutination. So this individual would be AB negative.
Oh, so all three samples are the same person?
All three samples are the same person. That's
Gotcha.
their blood that's put in here. For the A, we had the A antibody placed on it. For B, we had the B antibody placed on it. And for the RH, we had the basically it's the D antigen, the rhesus monkey antigen. And so that's how we determine our antibodies. We are.
So is that O negative? No, O negative.
Nope. This would be an AB negative.
AB negative. Okay.
Yeah. So what I'm going to ask you guys to do is you guys are giving these 3 samples, right? And I'm going to ask you, what is the blood type of these samples? So the first thing that you need to do is to go, so this is you, your right, your left, all right? And so on for the anti-A, if we go look at this for left, is there agglutination here? And, and you can get that picture a little bit bigger. I'm not doing the best at this. But on this one here, do you see, is there agglutination in this picture? The anti-A.
Looks clear. I'd say no.
Yes. No, and again, like this, when it's clear or it's hazy, that would be no. However, if it's like this, where it's all clumped together, that's a yes. Do you get that idea?
So on that one, on the last one you're pointing at, I only see agglutination on the 3rd circle, right? Is that correct? On the right?
I would say there's agglutination there too.
Okay, I think I'm gonna have to zoom these in then. It's kind of difficult.
You need to zoom them in a little bit. Would people need a little bit of hand on what blood typing? Maybe let's see.
It'll make it better, I think, if I like you did, make those images really huge. But I think I'm going to make it on its own separate row so that I can make the image bigger, because I think the table is making it not so you could make it the image huge. Oh, I know what I'll do.
All right. Are you guys just seeing the white page? So I got a new share. Do you guys see this? I just did an explanation of blood typing video, and there's a whole bunch of videos in here that explain kind of what blood typing is. If you need to look at, what I would definitely do is read the text, and then you could go in and you can do some of these and look at some more of these blood typing videos to kind of help you with that. Is that okay?
Yeah. Actually, I kind of like that. I don't know if you could see that girl where it says blood types easy 1515. Yeah, she's a registered nurse. I really like her content.
Okay. So if you've looked at
This is Sarah with registerednursern.com, and in this video, I'm going to be going over blood typing, specifically what a person can receive with a certain blood type. So let's look at these two. Okay, so what are antigens? Antigens are proteins that are found on the surface of a red blood cell. And depending on what type of blood you have will depend on what type of antigen you have on your red blood cell. wrong type of blood, and you get this antigen antibody reaction. Okay, so we have type B antibodies. Okay, type B, you're going to have to pay attention to the antigen on the surface of the red blood cells. Okay, so type A, they have A antigens. So we don't want to take their blood and give it to someone who has A antibodies in their plasma. So,
all right, did you guys see that?
Yeah, I saw that.
Okay, I could send in the link to hers in there, but there's a whole bunch of them.
They have
There's a whole bunch of them that are
in their plasma.
There's a whole bunch of them that are out there that you can look at that are all kind of equally well. As long as they're talking about what the antibody is and what the antigen is, that is great. Anyone have any questions?
I had a question. So without having a blood test, you can't really tell what your blood type is, right? Like there's no like home exam that you could do?
Not that I know of. This test is pretty easy. I mean, when we do it in the lab, it's a real easy test. All you need is you do a finger prick and you put it on a slide, and then you have the antibodies. So there's a pretty easy test. But again, on this test that we do, we tend to get a lot of people who are AB, and that may be due to our population, but I think it may be due to sometimes the test not being as accurate as I think it is. But no, for the most part, you're not going to have a test at home that you can do. What would you need an at-home blood test for?
Well, I'm just curious because I don't know my blood type, and that's something I probably should know. You know?
Yeah, I think at your doctor's office, if you just tell them and look at your look at what you have, because I'm sure, it's a pretty normal test that they do almost all the time. But sorry about that. We would, you would be able to know your blood test at the end of this laboratory, but we're not going to be able to do it. So,
if we ever get back to school this semester, could we do it? I'm curious. I want to know my blood
if we get it's a real easy test. We have everything all available all the time. So if you guys want to do a blood test when we get back, if we get back to school, when we get back to school, I have no problem doing it with you guys.
Cool. Thanks.
Okay. Does anyone else have any questions or anything?
Oh, I see Sierra. Sierra is just asking, when are labs due? 23, 32, etc. And
I'm going to have everything EVRI thing is due Sunday night. Everything is due Sunday night, midnight, before spring break. Is that okay with people? That will give me enough chance to kind of grade that over the Monday and still have the spring break and stuff like that.
Should we just work with what we have for the labs, or did you think we were maybe going to get more data on the root growth and stuff like that?
I will send you a note on Wednesday. That's Wednesday or Thursday. That's when Christian will be able to go into the laboratory, and I'll try and get him to take the measurements.
Okay, cool.
I at least wanted him to have a picture of the planaria. Actually looked really good, and we should have 4 totally new planaria on there.
Great, thanks.
Okay, anyone else? If that's it, I will be available. I will be available by email, cap@smccd.edu. If you want to, so I would strongly suggest that you go ahead and take about the next half hour and try and do this laboratory, and I will be here, and you can email me at smccd.edu till noon. So I will monitor this, and I will answer your questions kind of right away. You can also text me at 650-255-2364. Do this lab
Well, I see Melissa popped in a question.
All right, Melissa, the observation lab. The observation lab and this one is individual. The ones that we did before where we had groups kind of working together with growth and stuff like that, you can do those as a group. But I would like this lab, the blood typing lab, to be individual. I would like your, because again, you guys should not have been together. In no way was that intended at all. You guys should have all individually gone outside and done your field trips, practicing 6 feet distance between people. Actually, sorry, I put that for everyone. So do lab 30. You have a okay. So I will be hanging out till about 12 o'clock, and I am all your guys during this time. Other than that, you know, again, feel free to call me. But in the next 2 hours, hopefully, I'm monitoring this for you, and I will get right online for you. All right. Any questions?
Yeah, one more. I'm just trying to schedule my time. How about Thursday? Are we doing another lab, or what's going on Thursday?
Yes, Thursday we will have another lab. I'm trying to get something called Labster to work, where it's the virtual reality, and you can do. There's a number of labs that we can do for that. So I was hoping to either do the meiosis lab, or if that's not available, what we're going to do is we're going to look at pictures, and we're going to do the mammalian evolution laboratory. And so mammalian evolution would be that's lab number 29 on page 171.
Copy that. Thanks.
Again, on all these labs, I will record all of these. If you're not able to make it during the time, that's it. If you have a question for me, you could go ahead and answer it. This time here, because this is normally the time that we would be in lab, I'm kind of devoting to you guys. That if you have a question, I will answer that question first during this time period. Otherwise, I'm just, everyone's kind of question goes in the queue like it did before. All right, any other questions? All right, I hope you guys are all doing well, and I will catch you later.
Thanks, Dr. Cap. See ya.
Thank you.